All right, that was the pregame. Good evening. Good evening. Welcome to the first HudsonAlpha U event of 2026. And welcome to 2026. Lots of faces here that we haven't seen yet this year. My name is Kelly East, and I get the privilege of being the Vice President for Educational Outreach here at HudsonAlpha. And I'm thrilled that you are spending your Tuesday evening here with us and getting to spend a night talking about really fascinating science together for a little while. Tonight we're going to walk through this evening through a series of stories, and there are all kinds of stories that we get to tell you, things about human history, things about medical breakthroughs, thinking about food security. And so I hope that you've got your coffee, you've got cookies, and you're ready to go on a bit of a science journey together. Here at HudsonAlpha, getting to work in education here, we get a front row seat to the incredible scientists and innovators here at HudsonAlpha that are working day in and day out to help fuel discoveries and help write the stories of science, both in human health as well as in agriculture. And they are joined by tons of other scientists and innovators all across the globe that are also trying to tackle really big problems and come up with creative solutions to those problems using science. And tonight, we're going to get to tell you some stories that are connected to the work of HudsonAlpha, as well as other stories that are happening across the globe that we are simply fascinated by and cheerleading on being in the scientific field. And I've got a really special treat tonight because you don't just get one storyteller, you get two. And so Madelene, come join me on the stage, please. And let's give her a round of applause. So if you have not had the privilege of getting to know this incredible educator right here, let me introduce you to Madelene Loftin. Madelene is not new to HudsonAlpha. She's new to the HudsonAlpha U stage. But Madelene serves as our Director of Educator Professional Learning. That's a mouthful, but what that means is that Madelene and her team are teachers of teachers. They are responsible for designing and building and implementing programs and resources that teachers are using in classrooms and get to pour into those teachers and empower them, teachers in science classrooms across Alabama, across the country, and across the globe. And one of those resources that we are putting in teachers' hands every year is the HudsonAlpha Guidebook. There's a tiny version of it that you probably picked up as a magnet as you came in tonight. That's a magnet, that's not a book, you can't open it. If you didn't grab one, grab it on the way out. But that's a magnet with a QR that'll take you to the online version. But the guidebook it's a physical book or a digital version of a book that we build every year to bring science to life, to bring brand new cutting-edge stories of discovery, both to you guys and our community, but also to teachers and students, so that teachers can bring things that are way too new for their textbooks to the classroom and connect the things that they're teaching to the real-life discoveries that are happening in the world around us. And so that is a team effort, but Madelene plays a huge role in bringing the guidebook to life. And I wanted her to get a chance to join me in helping tell you some of the stories of the brand new guidebook that is just starting to hit the press. So we put out a new guidebook every March that covers stories that happened the previous year. So is it March? No, not quite. So if you scan the QR code, it's going to take you to the 2025 guidebook right now. Give us a couple days, and it's going to magically flip over to the 2026 guidebook, which is going to include the stories that we're going to tell you tonight, as well as a bunch of other stories that we don't have time to get to tonight. So we are right in the cusp of these things are really right hot off the press that we're telling you tonight. And we're excited to get to tell you them and for you to go explore them more online in a few days as well. Okay. Madeline, are you ready to dive in?
I think so,
okay.
And that was a great way for me to check and make sure this microphone was on.
So we're going to go back and forth, and I'm up first. The first story that we're going to talk through that was discovered this past year is not about people that lived last year. We're going to go back several thousand years and think about how DNA science is helping us understand ancient history. If you were at this talk last year, we also told an ancient DNA story about Mount Vesuvius and the people who perished in that eruption a long time ago. If you were here the year before, we also told an ancient DNA story, that time about head lice and about how tracking the DNA, I see some nods, yeah. About how you could track human migration across the planet over time by looking at the DNA of lice. If that sounds interesting and you don't remember it, go to that website and go to 2 guidebooks back and you'll find the story. But in this year's guidebook, we've actually put together an entire section about ancient DNA because there's more and more of these stories coming up, and we think they are just fascinating. But the story in this guidebook is not so much interested in the DNA of people that lived a long time ago. It's a story looking at the DNA of pests and germs that were living a long time ago. In this fascinating field called paleoepidemiology. It's a really big word, but it smashes together 2 words that you probably are more familiar with. Paleontology, right? Studying archaeology and the past, and epidemiology, which is the study of human health and diseases at a population level. And so putting those 2 things together is this field where you are not just looking at skeletal remains, you're not just tracking public health, but in this case, you're also bringing DNA into the mix because parasites, bacteria, viruses, they have genetic information today, and they had genetic information a long time ago, and you can learn about those viruses, bacteria, and parasites by looking at the DNA that is still around in human remains from a really long time ago. And that's what this story is about. So there was a paper published where scientists looked at human remains of more than 1,300 people over the course of a long period that were alive over the course of thousands and thousands of years and from a very wide distribution. Now, the scientists in this paper did not go find all of these remains. They took advantage of lots of studies that had been done and pulled data from lots of them together. And here's a picture of the geographic distribution of where all they looked at the DNA of these human remains. So we're all over Europe, all over Asia, and based on what we know about where people were buried, you can kind of get a sense of when they were alive, how long ago. And these numbers are hard to see up here. If you're in the back of the room, they might be more legible on the notes in front of you. But this shows when these people were alive. And it really spans everything back to getting closer past 10,000 years ago. So this is a really robust data set of a lot of human remains over a wide variety of space, over a wide range of time. Now, when they got the samples of these human remains, they've got DNA from a lot of different things. Certainly the person, and here they're getting the DNA out of bones and out of teeth. If you're into forensic files and you're thinking about exactly how they did this, but they certainly are getting the DNA of the person that died. They also, though, are getting the DNA of potential pathogens bacteria, viruses, parasites that that person was harboring at the time they died. But you can imagine they might also be getting DNA of other stuff too. Perhaps where the person was buried, there were other microorganisms or things living in the soil. So doing this analysis, they had to tease out, right, what's the DNA of people, what's the DNA of things that might just be environmental from where the person was buried. But they are able to find DNA that is at very high concentrations that matches known viruses and bacteria, things that make you sick, that make they're able to say, yeah, this person was actually afflicted with this at the time that they died. So they get the DNA and they sequence it. They read the DNA letters, and we're not talking about a little bit of data. We're talking about big data. So in this study, when you are sequencing DNA, even if you're sequencing DNA of someone who's currently still alive, you have to break the DNA up into lots of little pieces, read it, and then put it back together again. When you're dealing with DNA of things, of people and organisms from thousands of years ago, that DNA is already partly broken up for you, right? DNA is fairly stable, but it does degrade and break apart over time. But they break it up into lots of pieces. They sequence, they read those stretches of DNA. So in this study, there were 4.5 billion sequences of DNA that these scientists were dealing with. They put them together and they compare what they found to known genetic sequences of pathogens, things that are pathogens that make us sick today, as well as things that we know about what pathogens looked like back thousands of years ago. And let's take a little look at what they found. They found lots of stuff you can imagine in 1,300 people that we're not going to dive into all of the findings, but we'll cover some highlights together. One of the big things they found was this bacteria, Yersinia pestis. Yersinia pestis is most well known for causing the bubonic plague back in the 14th century. Now, that was not its first time causing a plague. That's the most famous. Yersinia pestis also was responsible for the Justinian plague back in about 500 AD that swept through the Mediterranean basin and killed lots and lots of people. But then it also caused the Black Death and then caused another plague in a much more modern era that started in Hong Kong in the 1850s and onwards. So this guy's been around for a while, and we've known that. We've known that Yersinia pestis has been causing plagues since 500 AD. But in fact, Yersinia pestis has been around a whole lot longer than that, based on what we see in these human remains that were studied in this paper. So in this map, you can see there's a really wide distribution of human remains that were infected with Yersinia pestis when they died. About 3% of all of the human remains that they looked at had Yersinia pestis. Over a wide geographic distribution, and they really got started right around 6,000 years ago. That's way before that Justinian plague. And this is fascinating because until this point, there's been a lot of thought that, probably before those plagues, there was some Yersinia pestis, but it was really caused by a few isolated zoologic spillover events where it moved from animals to humans. There wasn't really human-to-human transmission. Well, based on this data, we think there was quite a bit of human transmission back thousands of years ago, finding people that were buried together that were both infected with Yersinia pestis at the time of their death. And so it was far more ubiquitous than previously thought thousands of years ago. And this is another chart that's in the paper that is fascinating. So broadly, these spikes is how common it was to find Yersinia pestis in the human remain from really long time ago till, well, this was still a long time ago. But you can see marked where those two pandemics are. This is the Black Death, this is the Justinian plague, but look right here. There's actually an even larger spike in detection before either of those two, where it was even more common in these human remains. And this fairly well lines up with the time in history by looking at the DNA of the Yersinia pestis bacteria, that Yersinia pestis evolved. It gained a change in its DNA that allowed it to be transmitted by fleas. That before that time, it had much less ability to survive in the flea gut and have that transmission. So that happened right around here, where we saw this huge spike in detection within these human remains. Fascinating. That's not the only pathogen, that's not the only bug that was found in these samples. They also found really frequently, again, 2 or 3% of the people's remains that were looked at had Borrelia recurrentis found in it. This is a bacteria that causes recurring fevers that is transmitted by the body louse. You're welcome for me not putting the picture of the bacteria, not the louse, up on the screen really large for you. They also found a set of cases of Mycobacterium leprae. This is a very slow-growing bacteria that causes leprosy. They found in some cases. Of note, where they found these cases were in remains from Scandinavia, and another organism that can transmit and get Mycobacterium is the Scandinavian red squirrel. And so this Scandinavian fur trade is implicated in the spreading of this during periods, and that makes a lot of sense based on where they found remains with Mycobacterium leprae in it. They found parasites that cause malaria, Plasmodium, in about 9 cases that they looked at. And then they also, viruses also have genetic material. And so they were also able to detect viral illnesses. One that they called out in the paper was this toctinovirus. You've all heard of it. No, not really. No, it's a fascinating virus that apparently has been around for a really long time. It causes no human disease, that's why you've never heard of it, but 80% of the human population is infected with it. You're welcome. No, it doesn't cause any problems at all, but it's really common now, and it's been really common for a long time. But this is a fascinating paper, and it's a fascinating way to think about what we can learn about DNA. DNA from humans, but this isn't a story about humans, really. This is a story about pathogens. And overall, when you look at many of these pathogens that they're talking about in the paper, they're pathogens that are that have animals that are important to their story and about human transmission. And when you look across all of those pathogens and across history, you can see this spike, this growth of zoologic, animal-transmitted diseases that lines up in human history when pastoral migrations and when that kind of agricultural economy around animals and the moving of people that are caretaking animals started to happen. And so that's where you get the paleoepidemiology, right? You're laying what do we know about humans and human history on top of archaeology and on top of genetic science, and together you get a much better view of human history than you could get by just looking at one of those fields.
It's fascinating stuff. So let's switch gears a little bit and talk about type 1 diabetes. Individuals affected with type 1 diabetes, what happens is their autoimmune system attacks the islet cells of their pancreas. Those are the cells that produce insulin. And insulin is the hormone that regulates blood sugar. I'm sure you guys are familiar with that, right? Right? Now, there's about 9.5 million people worldwide affected with type 1 diabetes. And most of them have to take daily, or in some cases multiple times a day, insulin injections. But for a few of those people, even regular insulin injections don't regulate their blood sugar. They don't manage it well at all. And those people have a real problem if the frontline treatment that's available to you, if insulin is not regulating your blood sugar, then what do you do if you have type 1 diabetes? Well, one thing that's possible is you give those individuals an islet cell transplant. Now, those are the cells in the pancreas that produce insulin. Now, before you jump up and volunteer and say, I would like to help, could I be an islet cell donor? These pancreas tissues have to be collected posthumously. So, donor pancreases, or would it be pancreas? Donor pancreas, you harvest the islet cells by actually washing them out of the pancreas. And once you have those islet cells, that's this purple cell right here in the middle of the screen, you're going to take those, you're going to wash those cells, you're going to treat them if you need to, and they're going to be placed in the liver of a person with type 1 diabetes. Now, of course, you don't place them in the pancreas because in type 1 diabetes, the autoimmune system is already ready to attack islet cells in the pancreas. So the first thing they try to do is hide them in the liver. But these come from a donor. These are not islet cells that the body is going to recognize as my own tissue and my own cells. So individuals that receive this kind of transplanted islet cells, they have to be on autoimmune suppressants for the rest of their life. Now, if you Google autoimmune suppressant side effects, you might notice a few things. It has a lot of serious side effects, including worsening diabetes. Oh, so this is a problem. So what researchers said is there some way that we could get these cells, these implanted islet cells, in stealth mode? Is there some way that we could hide them from the immune system? And so that's exactly what they did. So they took some of these donor islet cells, and the first thing that they did was they altered them so that they're going to produce fewer cell surface markers, and basically they're going to put these cells in disguise. So that is a way for those islet cells to say to the immune system, I'm not a threat, I'm fine, I'm supposed to be here. And the other thing they did is they increased the expression of one particular cell surface marker, which serves as a big sign that says, don't eat me, to the white blood cells. Leave me alone and don't eat me. So now we have these altered islet cells. We ought to be able to transplant that into a patient, and that patient should be able to produce their own insulin. That was the next thing after they got these cells to grow in culture. And this is how that was reported in the news. Now, here is the actual paper. Now, let me tell you exactly what they did because it's absolutely fascinating. So they took these altered islet cells and they placed them in the muscle tissue in the forearm of an individual with really poorly controlled type 1 diabetes. So these implanted islet cells in his forearm, he did not get any immunosuppressant therapies. Those islet cells survived, there was no immune response, and they produced insulin. Right? That is incredible. This is a person with type 1 diabetes actually producing insulin. Now, this was a safety test, so it was a very small amount of insulin. They only put a few of those cells in there. They're trying to see, is there an immune response to these cells, and do the cells survive, and are they able to produce insulin? So this is a cautionary tale. This is amazing. If islet cell transplants can be helpful for those individuals with type 1 diabetes, in particular those that have poorly controlled diabetes, this could be a game changer. But we should not jump straight to the word cure.
Thanks. Okay, so that's islet cells, that's diabetes. We're going to stay in human health and in the realm of medical breakthroughs, but we're going to head up to the brain for a few minutes. And this next story is a story about a condition called Huntington disease. Yeah, I heard some, ooh, yeah, that's heavy. Huntington disease is a disorder of the brain. It's a neurodegenerative disorder that affects adults, where the brain is formed and it is functioning normally, and then there are changes that happen, and Huntington's leads to this constellation of symptoms that start, sometimes you don't even recognize them until you're looking backwards, but this progressive growing set of symptoms related to memory and brain cognition, muscle coordination and movement, and psychiatric issues that progress and ultimately lead to death 15 to 18 or so years after the time that symptoms onset. Onset's usually in the 30s or 40s or somewhere in that range. So it is a debilitating disease. It's one that once someone is on that course, there's very little that can be done to alter that course currently, other than help mitigate symptoms. There are changes, and I'm about to get into what is actually happening inside the brain cells that leads to these symptoms, but it's a buildup of a protein that damages your neurons and makes them die. And we don't regrow neurons really quickly or easily. And so neurons die and the brain ultimately shrinks and withers, leading to the symptoms of Huntington disease. Ooh, come back. Okay. But to think about what is actually happening, because to treat a disease, you've got to understand what is actually causing the disease inside of our body, inside of our cells. And Huntington's disease is a genetic disorder. There is a change in a DNA recipe that ultimately determines whether someone is going to develop Huntington disease or whether they're not. It is a classic disorder of where your genes completely control that question. And there is a gene called, you guessed it, it's the HTT gene, but it tells your body it's the recipe for making a protein called the Huntington protein. Makes sense. So inside, this is a really, really large model of a cell here, where you've got the nucleus, where your DNA is in the cell, so you've got a section of DNA that's the recipe that tells your body how to make this Huntington protein. So you've got DNA, and if you think back to biology class, you might remember that for DNA to help make a protein, there's this thing in between called messenger RNA. It's a working copy of a section of DNA that ultimately is used to guide making a protein. So this is your DNA up here that is ultimately transcribed. It's copied over into a messenger RNA. Messenger RNA is only one strand to DNA that's double-stranded. And that messenger RNA has the ability to leave the nucleus and go out into the main part of the cell where it is the code for making a protein. And in this case, it's the Huntington protein. So that's how all genes code for proteins. But in Huntington disease, what makes someone ultimately develop Huntington disease is their recipe is different. They have a recipe that has some extra information in it that you don't want. There is a portion of that recipe for the Huntington protein where there's these 3 DNA letters, CAG, that is copied over and over and over and over again. It's a copy and paste error where you've got too many CAGs. We all have a few CAGs at that spot in the recipe, but people who develop Huntington disease have too many. And what that means is then the protein that their body has an instruction to make, the protein is too long. It's got extra building blocks in it, and that is toxic to neurons. And when you look at the numbers here, what this looks like in practice is that if you were to sequence your HTT gene, you were to read your recipe, and you have 26 or less of those CAGs, you will not get Huntington disease. If you have 27 to 35 of those CAGs, you are not going to get Huntington disease. But there's an asterisk. But the next generation, it's unstable. The number of CAGs in your children might expand and be longer, and that it might push them over this threshold right here of 36. And 36 or more CAGs causes Huntington disease. So when we are testing people and deciding, is someone at risk of Huntington's or not, that's what we're doing. We're sequencing this gene and we're counting CAGs. And based on this, we can say yes or no for a particular person. So the reason we've just had a lesson in what causes Huntington's is that there is new research and a new study that was done this past year in thinking about using this knowledge to actually try to treat Huntington disease. Because if it is a toxic protein that is causing Huntington, how might you address that? Turn off the making of that protein, right? Because we have something there that we don't want. And so we are wanting to stop this process from happening potentially for the toxic version of the protein that's being made. And so the drug that has been developed and has been tested that I'm going to show you, or we're going to talk through in just a minute, is coming in at this stage and blocking things at the messenger RNA level. So it's allowing the messenger RNA to be made, but it's a drug that can come in and bind to that and stop it at that point so that the protein is not being made. So again, kind of like where Madelene was just talking about the word of caution, right? What I'm about to show you is a small-scale clinical trial where of a brand new drug called AMT-130. I'm sure they'll come up with a better name for it before it ever hits the TV commercial, right? And to ask your doctor about AMT-130 is not going to catch on, but we're not there yet anyways. But this is a small-scale clinical trial of this drug that is administered directly to the brain. And what the drug is it's a virus vector, meaning you're using a virus to get DNA that you're interested in to the place you're wanting it to go. But what they have inserted is a gene, is a DNA recipe for a microRNA that will recognize and bind to the Huntington messenger RNA and get rid of it. So this will allow those neurons to create a microRNA that will kind of search and destroy the messenger RNAs for Huntington. Yeah. And because they're not inserting the microRNAs, they're inserting the recipe for them. This is a treatment that could theoretically only have to be done once, right? You're inserting the recipe into the neurons so that they can continue over time to keep producing these microRNAs, right? You've got the recipe inserted, and so theoretically this could be a one-time treatment, which is good because you're doing brain surgery. So this is a small-scale clinical trial where they treated 29 patients with moderate to severe Huntington disease. So 29 patients, they had them broken up into 2 different groups where one got a little higher dose than the other, because again, this is clinical trials. They're trying to figure out dosing and safety and seeing what the right treatment is. And they followed them over 3 years and watched and monitored and how they were progressing, if they were progressing, and compared their progression to a whole bunch, 1,600 patients that were not treated, that are just out there being monitored. And what they found was that the treated patients had 75 to 80% less progression of Huntington disease compared to untreated controls. So that's really striking data. Now, that's not zero progression. There was still progression, but noticeably less progression for a disease that there are not good treatments out there today. So where do we go from here? With this particular drug, they've submitted that. They are in talks with the FDA, trying to think about, how can this get approval as is? What other trials need to be done to continue to look at this? But while that's a major breakthrough, there are some caveats and some asterisks to that. As not a easy treatment. Right? You are injecting through the skull into the brain. So that has challenges that come with it. Another downside to this particular treatment is that people who have Huntington's, they've got 2, and all of us in this room, we have 2 copies of the Huntington, the HTT gene. If a person who has Huntington's has that mutation in one of their 2 copies. So they have a typical, normal, working copy of HTT that is producing normal, typical, working messenger RNAs. This drug is going to non-selectively bind and destroy both of them. And so it's not selective. And in even this trial, there is still progression. This is not a cure, even if it worked as shown. And also, you can imagine that this is not a cheap process, which is true of many genetically informed treatments that are out there. That's not unique to this, but that is a challenge that will need to be addressed for this to be widely used. And also, a word of caution that we love to tell you these stories about incredible things that we're learning, but you have to stay curious, and sometimes things that look really promising don't pan out. And in a couple of years, we might have to stand up here and say, hey, remember that story we told you? Well, science kept doing science, and turns out what the scientists were seeing early on didn't pan out in bigger studies. This is a headline of a completely different drug that was also for Huntington disease that showed really promising early studies, and then when they did larger studies, it did not pan out at all. And they stopped the study early when they started looking at the data, that the people who were being treated were no better off than the people who weren't treated, and they had a bunch of side effects. So the hope is that AMT 130 will not have that same fate, but we are bringing you guys stories that are really early on, that are fascinating, but we are sitting in the process of coming alongside and watching these things pan out. And so we'll see. where this one goes.
And while we are on the tail end of cold and flu season, so viruses don't just impact humans, they also impact other organisms that we know and love and eat. So I'd like to introduce you to 3 new viruses. I'd like to introduce you to the porcine reproductive and respiratory virus, the tomato spotted wilt virus, and the cucumber mosaic virus. They aren't much to look at. But together, these 3 things are anticipated, they are expected that on any given year, if you look at the impact globally, they do more than $30 billion worth of damage every year. And they're not even all that fancy. But what we want to tell you about tonight are 3 really new, interesting methods to start trying to tackle these viruses. So the first one, let's dig into this porcine reproductive and respiratory virus. What this does is exactly what it sounds like. In adult pigs, it causes reproductive failure in stillborn piglets. In baby pigs, it causes respiratory symptoms that are really, really damaging. It causes fever, excess mucus in the lungs. What happens with a piglet that is infected with porcine reproductive and respiratory virus is it isn't the virus that kills the piglet, but it makes them vulnerable to opportunistic infections. So lots of piglet loss, lots of loss due to reproductive failure, and all because of this tiny, unassuming, not terribly interesting to look at virus. So let's talk about the way this virus gets into the respiratory cells in pigs. The virus there, which is got the little red spikies on it, what it does is it attaches to this cell membrane protein. This membrane protein acts as a channel. It's basically a tube, like a straw poked through the cell membrane. So when the virus docks, it takes advantage of that tube and uses that to get its genetic material inside the cell, and then that genetic material heads for the nucleus and takes over. So what researchers thought is, what if we did something to this mechanism that the virus is using to get inside the cells? So what they did is use CRISPR, and CRISPR is a technique that we've talked about multiple times in HudsonAlpha U and all the way back to Biotech 101 and 201. What CRISPR does is basically find targeted regions in the DNA and make specific changes. The changes that were made in this case, they removed 1 exon from one particular gene for this transmembrane protein. And what it did is change the shape. It changed the shape enough so that the virus can no longer bind to it, but the cell membrane protein actually continues to perform its normal function. So it still passes materials through the cell membrane, just not viral DNA. So that means that any pig that has been altered with CRISPR, while they can take the virus in, the virus cannot enter their cells. They are, in essence, a dead-end host. They can have the virus inside them. The virus can't reproduce. They can't pass the virus on to other pigs that are near them. So in 2025, late 2025, the Food and Drug Administration decided that these CRISPRed pigs did not pose any greater safety risk than eating pork from unaltered pigs. So what they said is this particular intentional genetic alteration, an IGA, this particular genetic change did not pose any greater safety risk than consuming pork products from unaltered pigs. So you may very soon see CRISPRed pigs in your market. Now, it is still up for debate. There is a crispy bacon joke in there somewhere, isn't there? There is still some debate whether or not the pigs that have been altered with CRISPR will require the bioengineered label. But the company that is responsible for this alteration in pigs has said they probably are going to use that label, whether it's required of them or not. Stay tuned, there's more to come. So now let's look at tomato spotted wilt virus. This is not a tomato. I know somebody was out there thinking, that lady doesn't know what a tomato leaf looks like. Anybody know what this is? These are leaves of a peanut plant. So the tomato spotted wilt virus is a challenge for lots of plants, not just tomatoes, and in particular for peanuts. And peanuts are really economically important plant in Alabama. So the tomato spotted wilt virus causes this pattern. They lose some green, some chlorophyll in their leaves. It makes these dead spots on the leaves. And of course, then there's reduced photosynthesis, there's reduced growth, there's reduced yield. Plants need to do photosynthesis. Is not good for a leaf. And they're carried by thrips. Thrips are tiny, tiny little insects that love to chew on new plants. So new growth on a plant are little baby plants. And that is how the virus gets passed from peanut plant to peanut plant. Now, the peanut plants are pretty hardy. They can survive a little thrip nibbling, but what happens when the thrip infects them with this virus, then they're infected with that virus for the rest of their peanutty life. Now, tomato spotted wilt virus was really devastating for peanut farmers in the late 90s and early 2000s, and a researcher discovered a gene that made some plants resistant to tomato spotted wilt virus. It didn't keep the plants from getting tomato spotted wilt virus, but it did mean that they were more likely to survive even if they were infected. Now, that's interesting because the way that you get resistance into plants, in particular in plants like peanuts, is you do the selection and the selective breeding based on phenotype. What does the plant look like? Somebody goes out into the field and said, that there is a good-looking peanut plant. And this peanut plant over here got tomato spotted wilt virus, but it didn't die. Let's cross the two of them and see if we can get that resistance into this really good growing peanut plant. Now, that process is incredibly laborious. That process, like, let me show you. This is how big the flowers of a peanut plant are. They're about 5 centimeters total. They are tiny. And if you want to cross peanut plants, it means you have to go out in the morning and remove the male part of the flower before it has an opportunity to self-pollinate. You have to collect all those. And then you have to go back in the evening, 12 hours later, and cross it with pollen from the plant you're trying to ingress. And you're trying to get those traits in there. So super laborious process. Well, researchers at HudsonAlpha have found 2 additional tomato spotted wilt virus resistance genes. So they found those genes by doing lots of whole genome sequencing of peanut plants and then looking at what are the functions of these genetic regions. Now, having identified these 2 new, so now we have a total of 3 genes responsible for tomato spotted wilt virus resistance. That's a mouthful. Now that we know that, we can use that information to make breeding decisions. So you don't have to wait until the plant grows in the field and say, oh, that's a good-looking peanut plant. You can actually do this when the plants are only about 2 weeks old, take a sample, extract DNA from that sample, test that for the presence of these 3 genes, and if they're there, you can decide which plants you want to cross before they even start producing flowers. Researchers at HudsonAlpha. All right, cucumber mosaic virus. Now, it ruins cucumbers, duh. But it also impacts a whole lot of other things that we like to eat, things like bell pepper and celery, all of the squashes, carrots. It even infects ornamental plants. So the pretty flowers that you plant in your yard can be decimated by the cucumber mosaic virus. And this one is carried by aphids. This is another one that's carried by insects, but it can also be spread by tools. If you go out and work in the garden in this part and there's some cucumber mosaic virus, if you take that same hoe over to another area in the garden, you're just helping to spread that. Yep. Now, what if you could give plants a vaccine, just like you guys might get a flu shot or a COVID vaccine? Could you give plants a vaccine? Well, yeah, we probably so. We wouldn't be talking about
This
it.
is a trusting room of people.
They really are. Now, our immune systems function very, very differently. For one thing, we have an immune system. We have a whole bunch of specialized tissues that are responsible for fighting off invaders. Plants really don't have that. Each individual plant cell has to be able to defend itself against invaders. So that makes vaccines for a plant are going to have to be something that work really, really differently from the vaccines that are in humans. But let's think about that. What if you could give a plant a vaccine? So what a vaccine does in people is it primes your immune system. It basically says to your immune system, here's who you're looking for. If this individual shows up again, attack them immediately. In plants, is there some way to prime their immune system? And what does a plant immune system do anyway? Well, what plant immune systems do is produce that. Trust me, you said it was a trusting audience. This is a lovely, spinning, double-stranded RNA. Trust me, it is. We only checked this 47 times today. This is a double-stranded RNA. Now, this is different from the messenger RNA that Kelly was talking about a minute ago because, first of all, it's double-stranded. It's much, much shorter than a messenger RNA. And what's important about this double-stranded RNA is this is how a plant defends itself. Plants make these double-stranded RNAs that can go out and attach to various regions on viral DNA. And if they hit just the right spot, they can shut down the infection process and block the virus from reproducing. So what researchers did is they said, hey, that's how plants work. They make these double-stranded RNAs. What if we gave it the double-stranded RNA that's going to work, that's going to attack cucumber mosaic virus every time? And so they did. They built a whole set of highly efficient double-stranded DNAs that they then inject into the leaf. This is from the paper. So what they're doing is they're pushing into the leaf tissue those double-stranded DNAs. And what happened when they did this in a research setting is that more than 80% of the plants that were injected with the vaccine survived when they infected them with the cucumber mosaic virus. Now, this probably doesn't have a practical application in agriculture. This would be labor intensive. But the hope is that researchers can take this, now that they have this as a proof of concept, when there really hasn't been anything except aphid prevention before now. Now that they have a proof of concept, the hope is that they can develop this into something that can be sprayed onto a field and thereby giving that resistance to cucumber mosaic virus. The other really nifty thing about this highly efficient double-stranded RNAs is that they can be made really quickly. So what that means is when the virus changes, when the virus mutates, as viruses do, that in a matter of weeks, researchers could retarget the mutated virus and actually have a new plant vaccine on the market really, really quickly. So 3 new ways to fight viruses.
All right, let's talk about why there's a slide full of gum on the screen. This is a story about a potential avenue for using gum to detect the flu. There is a real strong virus undertone to a bunch of these stories. But yeah, you heard that right. Chewing gum to detect influenza. That seems wildly far-fetched, and it's actually not quite as far-fetched as it sounds. So the background here is, so influenza it's a virus, just like the story Madelene was just talking through about how viruses get into cells. This little graphical animation here. There's your spiky virus and the cell that it's trying to get into and hijack, and it's going to make its way into that cell, and it's going to spill its guts right out into the cell, head into the nucleus and hijack the cell's ability to copy DNA and make copies of that, to copy the DNA or RNA of the virus, to make a whole bunch of new virus particles that then will leave the cell and go find some new unassuming cells to go do the exact same thing inside. So that is your virus life cycle, incredibly simplified. But there is a critical feature to this whole process, and that is an enzyme that the influenza virus and other viruses as well have. They've got a recipe in their genetic material to create the neuraminidase enzyme. This is an enzyme that the virus is making that is important for this process right here, getting the viruses back out of the cell, releasing them to go find new cells to infect. This enzyme helps break or cleave bonds that are attaching and keeping the virus, the new virus particles, stuck to the side of the cell, right? It releases that so they can leave and go off and infect new cells. So neuraminidase enzyme is an important enzyme, but it is only present when there are, in this, viruses around, because it's being made by that virus. And so what some really savvy, creative scientists have done is they've built a taste-based sensor that would detect influenza being present based on whether or not this enzyme is present. So this is not a perfect, highly sensitive sensor here. It's a yes, no, is this enzyme there? And if the enzyme is there, you're making an assumption that that's because there is virus around. But the way this works is they've got, I will get off this slide quickly and not sit in organic chemistry here, but this is the molecule of the sensor that they made. So it has this acid, and they have attached a thymol molecule to it. Thymol has a taste. And so if this sensor is in the presence of virus particles that are making that enzyme, it's going to break that apart. It's going to cleave that, and that's going to release this taste molecule over here for a person to be able to taste with their taste buds. And thymol is kind of warm, spicy, woodsy flavor. It's found in thyme. It might be in your spice cabinet. It's a detectable taste. And so, but you don't taste it unless that molecule has broken in the presence of this enzyme made by a virus. So that taste sensor could go on a lollipop. You could put it in gum. You could put it in a taste strip, right? You can imagine all kinds of applications for that. Now, bacteria also create this enzyme, but these scientists were savvy and able to have this sensor built so that it can differentiate the two, that the bacteria version of the enzyme will not make that break when the viral version will. So this is a graphic that I pulled just a week or two ago from the World Health Organization showing that for the previous week, this is a constantly moving target, but the current read on how things are going with influenza in the world. We all know how influenza is currently going in our area here, and there's a lot of it, right? But that is true all across the globe. And this, we're not saying that doctors are going to start using a lollipop to determine if you have the flu in the doctor's office instead of a more sensitive test, but you can imagine the utility of something like this in terms of quick checks or in areas where fast cost-effective approaches to being able to detect the potential presence of influenza, where you could get potentially people to quarantine and stay away faster, right? And then maybe follow that up with more specific testing. But it has the potential to be a pretty big game changer from a global standpoint of a fast, easy, cost-effective way to detect the flu. It's pretty cool, right? Yeah.
So there are no fewer than 2 biology teachers in the room. What do we have before us? Cells, beautiful. We've got a plant cell and we've got an animal cell. Regardless of when you went to school, at some point you had something that you had to label these parts on. Somebody right now is thinking about their very favorite cell labeling color sheet. We're all pretty comfortable with this. We know that that big purple blob in each one of those is the nucleus. That's where the DNA is. We're pretty sure that these green things over here on the plant cell are the chloroplast. You could probably pick out the mitochondria and tell me it was the... See, there you go. So you feel pretty good about cell organelles, those internal structures, those small compartments that do a function inside a cell. Most of us feel pretty good about those. Well, don't be too comfortable, because I would like to introduce you to a new organelle, a newly discovered organelle. This organelle is called the nitroplast. There is a big giant green nitroplast. It is surviving in an algae, marine algae. Now, this nitroplast has a really nifty trick, because one of the things that it does is it fixes nitrogen. Now, those of you that remember being in school probably heard your biology teacher tell you that only bacteria can fix nitrogen. We know about bacteria that live in the root nodules of legumes, right? That is why we know George Washington Carver's name and the peanut plant is so important, is because peanuts are legumes. They have these nodules on their roots. The bacteria that live in those nodules are able to capture nitrogen and store it and put it in the soil so the peanut plant can absorb it. All living things need nitrogen. Nitrogen is a key feature of all of the proteins our bodies need to be able to make. All living things need nitrogen. So the idea of something living inside something else and being able to fix nitrogen is not particularly new. The difference is, this is a situation where it's symbiosis. You have bacteria, which are also alive, living inside the roots of these plants, living inside the root nodules of legumes. If you took the bacteria out of those root nodules, the bacteria would be like, cool, I was happy there, but I can move to a new apartment. They're not worried about that at all. That's very different from what's happening here in the nitroplast. And actually, the nitroplast was discovered in 2024. But we wrote about it for the guidebook this year because it was not until 2025. This was a hot debate in the scientific community. Is this just a case of symbiosis, and that is a free-living bacteria inside the algae, or is this a case of an actual new organelle? Hot scientific debate, which basically means technically worded, peer-reviewed papers and politely phrased letters to the editor. But in 2025, they got enough evidence to say, no, without a doubt, this actually is a new organelle. So these are electron micrographs of the algae, and the new organelle, the nitroplast, is in this kind of bluish teal color. So what they found is that the nitroplast grows with the algae, it divides with the other organelles inside the algae cells, and it divides with the algae, and it has a dramatically reduced genome. So basically, this remnant of what was once a free-living bacteria that could grab nitrogen, this remnant has lost most of the genes that it would need to be free-living. It now depends on this algae cell to provide it nutrients, to protect it from desiccation, all of those things. So without question, this is a new organelle. This is a case of endosymbiosis. And what that means, biology teachers, is you're going to have to get new worksheets. Because imagine now we're going to have to add these teal-colored organelles onto all of the plant cells in there. What is super interesting about this is because this is not a single gene, this is an organelle, this is a whole cellular membrane-bound structure. Imagine if you could put this organelle inside other plant cells and not just this algae. Then what you would have is a plant that made its own fertilizer. Yeah. Yeah, it might be worth getting a new color sheet.
All right, so for our final story tonight, let's talk about baldness, shall we? So I don't need to explain to you what baldness is or how many people it impacts, some by choice and some not by choice. You've been down the aisle in the pharmacy. This is not like Huntington's where I say there are no treatments currently available. There are all kinds of remedies and treatments that have been developed to stimulate hair growth. But this is a story of another one getting added to that tool belt and being tested that is really interesting and showing some real promise. So our hair grows through a cycle that you have periods of hair growth and hair rest, and it kind of goes in this cyclical structure that is all being directed by chemicals and signals from the hair follicle stem cells that sit below where hair would be growing. And when you have hair loss, that can be caused by a lot of different things. You can have a lack of hair growth because of damage, where there is actually, where the hair follicles, that has been replaced by scar tissue, and that is fairly irreversible, right? You don't have the hair follicles there, the stem cells, to restart things. But there's lots of situations where the hair follicle stem cells are there, they're just resting, they're dormant, they are not sending the signals that would create hair growth. And so that's where there's a lot of work trying to figure out, is there a way to tell them to turn back on when they are turned off when they shouldn't be. And so scientists at UCLA led this initial work. They've discovered a small molecule that they call PP405. So this is a small molecule that targets a protein that is in those hair follicle stem cells. And adding this small molecule essentially wakes them up. It turns them on. It tells them to send the signals to grow hair. This is not a really deep story to end on, right? Fairly straightforward. But so they discovered the small molecule, and so they have tested it. In 2025, there were phase 2 trials that happened where they tested it in 78 people, and, well, they grew hair. So it worked. And so looking ahead to 2026, they're about to start a whole nother set of phase 3 trials where, they're moving closer to potential FDA approval. And so perhaps coming to a pharmacy shelf near you sometime, not in 2026, but maybe at some point. And again, it's probably not going to be called PP405. Come up with another name. But perhaps, the hair growth that they're seeing with PP405, they're really excited about, that it is not peach fuzz that is growing, that it is longer type hair that is being stimulated and grown with the addition of this small molecule that is rubbed on topically. So there you go. The headlines that came out from this was something about scientists have cured baldness. Again, not quite there yet, but is really fascinating. So that was a set of, I don't know how many stories. There was the one you did that had several smashed together, but upwards of 10 stories in the guidebook for 2026. That was a few of lots of really incredible science that we have written about. So I hope that after tonight, you give us till March, when we said that we will populate the new guidebook online, that you can go and read about these stories as well as so many more stories that we don't have time to chat about tonight. But all of the back issues of the guidebook are up online as well. But with that, we will close in just a minute. Let me point you as well. Coming this fall, so this is Hudson Alpha U event. Ooh, come back. Coming this fall, we're going to do a whole 3-month series. So there will be a Hudson Alpha U every month in September, October, and November about the science of healthy aging. And that'll be myself and Dr. Neil Lamb that are going to co-lead that together. And so that's going to be really fun. Stay tuned for more information about that coming up later on this year. As you leave tonight, in the next couple of days, we'll send you an email saying thank you so much for coming. It's also going to talk about the Hudson Alpha Alumni Association. The Alumni Association is our family. It's the people who attend our events, you guys that are here tonight, students, teachers that come to our events, that we like to stay in communication with. So by coming, you are part of our Alumni Association, but it's also our way to stay extra engaged, send updates to you, and ask for your support. So in that email that you get, you'll see a invitation to join the Alumni Association at a silver or a gold level. We would absolutely always love your support in the things that we do in education to put these events on, as well as the things we do for students and teachers all across our community and our state and our country. So keep an eye out for that. And then finally, we would not be here tonight in this beautiful space with those wonderful cookies that I'm not seeing many out and about now because they're in your bellies. We would not be here without our sponsors. So a big thank you to the Ashburn Foundation, Longview, and the Jackson Center for coming alongside our mission to keep the community engaged. So with that, I just want to say thank you again for spending your evening with us, braving the cold and the dark. Before we go tonight, Madeline, come over here, and y'all know I tend to take selfies from stages, and so we're going to do that real quick. Okay, all right. All right, so thank you, Madeline, for joining me tonight. Isn't she a marvelous storyteller? All right, with that, we're gonna hang out down here. If you wanna chat more about any of these stories or anything else, we'll be down here, but y'all have a great night, be safe, and we'll see you back on Hudson Alpha's campus soon.